Hello, 7th grade. Today we are learning about something new. We're learning about how to predict genetic outcomes using what we call Mendelian Genetics and using this tool called a Punnett square. So by the end of this lesson, you should be able to use a Punnett square to determine genotypes and phenotypes of certain organisms. All right, moving on to the next slide. All right, so just to summarize what we learned about yesterday, using our strawberry lab, you should have taken away two things. One is that every single organism contains DNA, which is genetic information. And that genetic information found in DNA is going to determine everything, well, a lot of things about that organism, including its genotype, which is the code, and the phenotype, which is the physical characteristics. So for example, a genotype for the strawberry could have been like RR, and the phenotype would have been the strawberry is red. So this is a kind of blueprint. DNA, when you see DNA, you always think that it's a blueprint of like how to create an organism. Now, DNA is bundled up into chromosomes, which you find in your nucleus. In order for us to have seen the DNA of the strawberry yesterday, we had to break down the nucleus and release those chromosomes from the nucleus. And we did that by breaking apart the strawberry and dissolving the nucleus. All right, some other things that we learned. DNA is like a twisted ladder, and each rung of that ladder has a base pair, which we call nitrogenous bases, but you just need to know that they are called base pairs. And the way that we remember what pair pairs up with whatever pair is always trust good cats, because if you split it down the middle, adenine always pairs up with thymine, guanine always pairs up with cytosine. So my DNA, if I split that in half, I would see that it would always go something like this. They would always match up with each other. So we could like pretty much split DNA in half and make like half of a ladder like that, which you're gonna learn about more in like higher level biology classes. All right, continuing our review. Let's do a quick check for understanding. Match the following DNA sequence to its base pair. So here we have a DNA sequence and we want to match it to the correct base pair. My first step is I always write, always trust good cats. Split a line down the middle, and that's how I know how I'm going to pair it. So A matches with T, C matches with G, G, C, C, G, T, A. Now all I'm doing is getting that from here. So the correct answer would be B. All right, so continuing to summarize what we learned in the strawberry lab, everything has DNA, and all living organisms have DNA, and that DNA is going to determine physical traits of that organism, like, for example, the color, the size, and the texture of that organism. So that's the DNA that we find in a strawberry, and you notice how they have base pairs over here too. It's going to determine like whether strawberries can be big or small, red or pink, etc., etc. Now, point number 2 is that unlike humans and other animals, strawberries reproduce asexually, meaning that genetic information from just one parent is cloned so that all of its kids turn out to be pretty much identical to the parent. So in this example, like you'll see that this strawberry, just one parent can produce more offspring by itself just by pretty much cloning itself through tubers. So what this means is that their kids are exactly the same. There's no variation. So for example, with humans, we do have variation. We look very different from our parents. But because strawberries reproduce asexually, they will look exactly the same as their parents. They are basically clones of each other. All right, so let's go ahead and begin with our lesson. We're gonna learn about something new today. So I just wanna introduce you guys to this guy first called Gregor Mendel. He is a very old dude. He lived in 1820s, in the 1800s. And he's really important because he's pretty much the first scientist who developed the principles of heredity. He explored genetics and how genes are passed on from one organism to the other. He was the first scientist to ever explore that. And he did that, like, he discovered this whole field of genetics without any knowledge of genes or chromosomes. He basically came up with everything from scratch, which is amazing. And he did that simply by studying pea plants. That's right, pea plants, like from like these kind of pea plants, yeah, the ones that we eat and see in gardens and stuff. He basically no, he was a monk, and he like had this garden of pea plants, and he basically noticed that like if I cross certain types of pea plants with each other or make them breed, there's always like they will most likely turn out a certain way, and that is because of genetics. Pause right here. Okay, a little bit of technical issues, but I am back. Anyway, back to Gregor Mendel and his peas. So basically, Gregor Mendel was like, oh my gosh, if I noticed over time that if I continue crossing purple pea plants and white pea plants, it turns out most of them are going to turn out purple. I wonder why that is. Maybe it's because one of the parents was purple. Or something like, okay, I crossed some green pea plants with some yellow pea plants. It turns out most of them will turn out green as well, even though some are yellow. I wonder why some of the pea plants kids are green and some of the pea plants kids are yellow. Well, because of genetics, because parents pass down their traits from themselves to their offspring with variation. And that makes a lot of sense to us right now, but back in the day, that was like it was like an amazing discovery. And so I'm not going to go super specific into like how Gregor Mendel figured this out, but he figured it out with the help of something called a Punnett square. And we're going to practice what that is right now, because what Gregor Mendel found out is that if genetics works in a very predictable way, like with certain tools, and if you know the genotypes of two organisms, you can cross them and you can pretty much figure out what their kids are going to look like or be like. And I will explain that in the next slide. Here we go. Oh, okay, here we go. All right, so Mendel basically, what he did was he cross-pollinated certain plants with different types of traits to see what would happen. And like basically by crossing certain plants, that means he breeds two plants together, and using this thing called a Punnett square, which you'll see over here, turns out Mendel was able to successfully predict what these two plants' kids would look like. So I'm just going to show you how he did it for now, and then we're going to practice doing it. So for example, Gregor Mendel took a really tall pea plant. There's the genotype, capital T, capital T. And then he took a really short pea plant, little t, little t. Those are their genotypes, right? We know that because they're codes. He took those two genotypes, he cross-pollinated those plants, aka made them have little plant babies, and he used this object called a Punnett square to predict how exactly their kids would look like if they had 4 kids. What he did was he lined up their genotypes right here. So for example, our really tall plant goes over here, and then our really short plant, he put their genotypes right here, right? Big T, little t, little t. And then what he did was he crossed them. So he took one allele from the top parent, so that's capital T, and then a little t. And then he did the same thing in the next box. Big T, little t. And then in the next box, big T, little t. And then in the last box, big T, little t. So he's basically just carrying down and carrying across the different genotypes of those two plants. And so what happens is, I'm going to kind of clear this up for you a little bit, just kind of clean it up. What happens is that he was able to predict the genotypes of their children. So if those two pea plants had 4 children, their genotypes would look like that. And it turns out that because, like, using this simple technique called the Punnett square, just crossing those genotypes downwards and across, you can basically see what exactly their kids are going to look like. So most of those plants ended up being really tall because of this capital T over here. All right, let's go to the next slide. Why did those plants all end up tall? Well, one thing that Gregor Mendel discovered is the law of dominance. Dominance, right? Dominance with a capital D. So dominance tells us, why is this not working? Here we go. Dominance tells us that whenever we have a capital letter, anytime we have a capital letter, even though there's going to be a lowercase letter, the capital letter will always win. So I'm just going to write that over here. Capital always wins, or it always dominates. So for example, let's fill out this Punnett square over here, right? T is tall, little t is short. And I know that because of this key right here. Turns out that tall is dominant, so I'm just going to mark that as dominant, and short is recessive. And then I'm going to fill out my Punnett square. I'm going to take the top t and the little t. I'm going to do the same over here. It's also a big T, and it's also a little t. Now I'm going to move to my next box, big T, and oops, and little t. And then over here, big T and little t. And I'm just getting those letters from the sides. Now we have the genotypes of 4 kids. Let's look at one genotype right here. I'm just gonna box this one. We're gonna look at this one right now. So I see that there's a big T and a little t. Knowing that there's a law of dominance, I know that the big T is going to be really strong. Capital always dominates. So even though there's a little t, I can basically ignore it. That big T covers up that little t because it's dominant. So because the big T is dominant, this kid is going to be tall. Now let me look at the next child. So the next pea plant that they crossed, they had a second plant child, and let me look at its genotype. Well, it has one big letter, and I know that capital always dominates, so this big T is going to overshadow the little t. So that means that it will always show the most dominant allele. So this kid is going to be tall as well. So it's going to be the same case with all the other kids. There's a big T, it's always going to overshadow the little t because capital always dominates. And so we know that all of the pea plants are going to be tall, which is great for the farmer. He has very tall pea plants making lots of peas, which means he has a happy farming life. And that's basically how you do a Punnett square with a law of dominance. We're going to do a bunch of practices of that in the next few days because it is a pretty tough concept to grasp. But basically, if I know like what the two parents are, I'm just going to clear this all out. It's all messy. If I know what my parents are, so here's my the mom plant, and here's the dad plant. What this Punnett square tells me with Mendelian genetics is I can basically just predict what my kids are going to look like. So let's go ahead and do some more practices with that. Before we practice, let's review some terms that you're probably going to come over in a bunch of these questions. Homozygous means the same, so I'm just going to pick a random letter here, D. So homozygous would be either capital D, or little d and little d. The point is, the two letters are the same. Heterozygous means different, so that means one big, one small. They are different. Dominant means that it's always capital. It's a stronger trait. Capitals always dominate, so that means this. And recessive means a lowercase weak trait. Lowercase They're very weak. They cannot stand by themselves, so they always have to have a pair. That's a recessive trait right there. So those are just your terms to go over. Let's go ahead and do this practice. So for each genotype below, indicate whether it is a heterozygous or homozygous trait. And this is when scientists went to Bikini Bottom and investigated the genetic makeup of organisms in this community. So if we went through Bikini Bottom and took all of the different organisms in Bikini Bottom and analyzed their genotypes, we found these genotypes. And we're going to practice our whether we know what a heterozygous or a homozygous trait is. So I'll get you started with a couple of these. Here we go. Big T, big T. These are the same two letters in the same capital way, so this is homozygous. Big B, little b, those are different, so they're heterozygous. Big D is the same, so that's homozygous. Go ahead and try big F, little f by yourself. You're right, that is heterozygous. What about little t, little t? You're right, that is totally homozygous because they're both small. And little d, those are definitely homozygous because they're both small. We're going to skip the rest of them because I'm pretty sure you guys get it by now. Okay, here it is. Let's practice if we know what dominant and recessive is, because you're going to really need to know this before trying your own Punnett square. All right, so determine the phenotype or the physical characteristic based on these genotypes using information provided about SpongeBob. If we're looking at sponges, yellow would be dominant to blue. So if yellow is dominant is always capital. And that means that recessive is blue, would be little letters. So for example, I see two capital letters here, capital Y's. Those are both dominant traits, so I can predict that it's going to be yellow because yellow is dominant. Big Y, little y. So this is heterozygous. There's a big Y and a little y. But because of the law of dominance, I know that the big Y is going to overshadow the little y, and it's still going to be yellow. And go ahead, little y. So I have two recessive, very weak traits. But since they're standing together, they're going to be pretty strong too. So this one actually means that this sponge is going to be blue. Now, go ahead and try how you know square. Go ahead and try to see if you know whether square is dominant to round. In this problem set, the square shape is dominant. So that means that square shape is dominant, so it's going to be capital. And then little s is going to be recessive. It's going to be little. So go ahead, try S, big S, big S. So that shows you a homozygous dominant trait. That means that he's going to be square. What about big S, little s? So again, law of dominance states that this big S is going to overshadow the little s, so he's still going to be square. And lastly, little s, little s. Okay, we have two very weak traits. They're recessive, but since there's two of them, you can totally see it this time, so this is going to be round. That's a little bit of practice. Again, we do need to know the law of dominance when it comes to Punnett squares. All right, so now we're going to combine all of that knowledge and we're going to tackle our own genetics problem for the first time. And I have three very simple, but you need to follow these steps in order to get it 100% right every single time. So here are the three steps. 1, write the key. What is dominant and what is recessive? Step 2, you're going to determine both parents' genotypes first. Genotype, of course, means the code. And then step 3, finally fill out your Punnett square. Let's go ahead and I'll do one and then we can do it together and then you'll do one on your own. So let's go ahead and read this problem. Patrick meets Patty. So Patrick meets Patty at a dance. Both of them are heterozygous for their pink body color, which is dominant over a yellow body color. Create a Punnett square to show the possibilities that would result if Patrick and Patty had children. Okay, so Patrick had a great night out and met a different starfish named Patty, and both of them are heterozygous. So first thing I'm going to do is I'm going to write the key. What does that mean, Miss Aquino? Well, the key is simply for you to remember what letter corresponds to what trait. So in this case, they don't really give me a key, so I can just make one up. So, all right, they're both heterozygous for pink and yellow, pink being dominant. So I'm going to write my key and say that since pink is dominant, I need it to be a capital letter. So I'm just going to do capital P equals pink. And I'm going to write also that this is dominant. And so that means that little p would mean yellow, and that this is recessive. And I just write that so that I never, ever, ever get confused. All right, so step 1, I wrote my key. Step 2, determine both parents' genotypes, so the code. So now the parents. So Patrick, let's write his code, and let's write Patty's code as well. So Patrick, it says here that he is heterozygous pink. So heterozygous means that they have to be different, capital and lowercase. So since Patrick is dominant pink, I'm going to do big P and little p. And since Patty says both of them are heterozygous, Patty is also heterozygous, so she has a capital P and a little p. I just got that from reading the problem. So now I have my parents' genotypes. Finally, I can fill out my Punnett square. So I'm going to draw my Punnett square like this. And I'm going to write my parents' genotypes on the sides. So Patrick was big P and then little p. Patty was also big P and little p. And so I am finally going to fill this out. So I'm going to go like that. So that's big P and big P. And then I'm going to go like this. That's little p and big P. I always like to put my big letters in front. And I'm going to remember that this is a little p. And then I'm going to go to this next box, take it down from the top, so big P from the top. And then from the side, I'm going to get a little p. Excuse me. And finally, my last box, I'm going to take the little p from the top and the little p from the side. And it turns out that this box has 2 little p's. And that's it. I've filled out my Punnett square. So those are the exact steps that you need to take to be able to fill out the Punnett square. 3 steps: write the key, determine the genotype, and then fill out the Punnett square. This would be your final answer. And that's how you do a Punnett square. So let's go ahead and try to do another one together. All right, so SpongeBob meets Sponge Susie. SpongeBob SquarePants recently met Sponge Susie Roundpants at a dance. SpongeBob is heterozygous for his square shape, but Sponge Susie is round. Create a Punnett square to show the possibilities that would result if SpongeBob and Sponge Susie had children. So go ahead and write your key. So, let's say that square is, since square is dominant, square is going to be big S. And it's dominant. So what would be the letter for round? Would it be capital or would it be little? You are right. It would be a little tiny s because this one is recessive. So let's go ahead and do our, well, I'm actually going to do that on the side. Where's my eraser? Let's go ahead and do parents. So SpongeBob and Susie. So it tells me here that SpongeBob is heterozygous. That means that his genotype would be 2 different letters. Go ahead and choose which one would show that SpongeBob is heterozygous square. That's right. SpongeBob would have a big S and a little s because he's heterozygous. He needs to have 2 different, has to be capital and little, but since he's square, he still has at least 1 big S. Now, Sponge Susie is round. We know that round is recessive, so which one would correctly show Susie's genotype? So Susie actually needs to have 2 little s's because she's round. It's recessive, and recessive means weak. Those genotypes cannot be by themselves. They need to have a partner to strengthen that genotype. So Susie has 2 little letters to show that she's round. And so I'm gonna write that here just so that we don't get confused. Susie is round while SpongeBob is square. All right, so we did step 1 and step 2. Let's finally do step number 3. We're gonna draw our Punnett square, and then I'm gonna write SpongeBob and Susie's genotype. So SpongeBob has a big S and a little s. Susie has a little s and a little s. And now I am going to draw. I'm going to finally fill out this Punnett square. So I just start from my first one. I take from the top and I take from the side. So big S, little s. My next box I take from the top and from the side. So little s and little s. Your next box, take it from the top and from the side. What belongs there? That's right, big S and little s. And go ahead and fill out the final box by yourself. Take from the top and take it from the side. That would absolutely be little s and little s. And that's how you fill out the Punnett square. So as you can see, let's go ahead and look at the laws of dominance here. So the top one would be, since there's a law of dominance there, since there's a capital, that child would still be square. And let's go over here. Since there's a dominant S, we're going to cross this out. So this child would also be square. Excuse me. This box over here, we have 2 little s's. And 2 little s's means that this child is actually going to come out round. And this box also has 2 little s's. So this child is going to be round. So it is possible for SpongeBob to have round children if he just meets the right type of girl. All right, so moving on. Let's go ahead and try one last problem together. So a heterozygous tall plant crosses with a homozygous short plant. How many kids will be short? Interesting problem. We have to specifically find out which kids are going to be short. So let's go ahead and start off by always writing our key. So heterozygous tall. So tall is probably, let's look over here. Okay, tall is right there. Tall is dominant. So big T equals tall, and I'm going to write that that is dominant. Little t is going to equal short, and that's going to be the recessive trait. So let's go ahead and do our parents. So parent number 1 is heterozygous tall. Heterozygous tall. What would heterozygous tall look like? That's right, heterozygous tall would be big T and little t, because heterozygous means they have to be different, although he's still tall, so he needs to have that big tall gene. Parent number 2 is homozygous short. What would it look like if you had a homozygous short plant? That's right, in order for something to be short, it has to have the little t, and since it's homozygous, they're the same. So it would be little t, little t. Now we're going to go ahead and draw our Punnett square. And then we're going to add our parents' genotypes. So parent 1 is big T, little t. And parent 2 is little t, little t. Go ahead and cross this first box. Yep, that's big T and little t. Now cross the second box. Little t and little t. My third box would be little t and little t. And my last box, what would my last box be? That would also be little t and little t. Awesome. Now that we have figured out our Punnett square, we also have to figure out which of the kids are going to be short. So look back at your key. Little t means short. So we have to count which boxes have little t's. Now, not all of my boxes with little t's are going to be short because, right, like for example, over here, we see that there's a little t, but that doesn't mean that that plant can be short. Since there's a law of dominance, this basically crosses this, the big T overshadows the little t. So this plant is going to be tall. This plant has a big T, so law of dominance, it's also going to be tall. But these children, they have 2 little t's. So how many of my plants are going to be short? That's right, only two plants can be short because these 2 plants have little t, little t. They're recessive, but they do have that short gene just like parent number 2. So that's how you would find out how you could predict which kids are going to be like what. All right, next slide. All right, so to summarize, we found out that the scientist named Gregor Mendel studied genetics for the first time using pea plants. He created the law of dominance, which just means that dominant alleles, or the capital alleles, will always show over recessive alleles. So even if my allele looks like this, my dominant allele is always going to overshadow that little one, so it's always going to come out no matter what it is. Since this is capital, law of dominance says, nope, I'm going to overshadow you. Even for this, even if you see that little one, you got to notice if there's a big one because it's always going to overshadow it. That's the law of dominance. We can then use a Punnett square to predict the genotypes and the phenotypes of offspring. And the steps that we take to write the Punnett square is step 1, write your key. Always write your key. Tell me what the big letter means and what the little letter means. Step 2, determine the parents' genotypes. And then step 3, you can finally fill out the Punnett square when you know the parents' genotypes. And I do know that, yes, today was very confusing probably, but don't stress out too much because we have 2 more days to cover Punnett squares. So we'll get more and more practice before our next test. Good luck. Go ahead and finish your worksheet and text me if you have any questions. That's it.